Okay, good evening, everyone. Can you hear me? The back? Great, okay. So, yeah, good evening, everyone. So my name's Rod Downey. I lead the polar program for WWF based in the UK. And I'm joined by two fantastic speakers and scientists who are joining online. That's Dr. Simeon Hill and Dr. Kate McCarry. And we'll introduce them in a bit. But first of all, I wanted to say a huge thank you to ICCI for hosting us at the Cryosphere Pavilion this evening for the launch of the first phase of a new science project. It's called Krill from Space, so it's definitely the coolest event title here at COP29. It's a collaboration between WWF, the University of Strathclyde, and the British Antarctic Survey. And we're also really grateful to Hurtigruten Expeditions, who are providing logistical support for the fieldwork that we're doing. So thank you to those of you who are joining us here in Baku in Azerbaijan. But thank you also to those of you who are joining us remotely around the world as we live stream this event. But you know, no matter where you are in the world today, it's very clear that what happens over these 2 weeks at the Climate COP in Baku, which of course follows very closely on the heels of the Biodiversity COP in Cali, is absolutely critical for the future of nature and the future of humanity. We know that our planet is on the brink of irreversible change. You know, over the last 50 years, we've destroyed nature, we've devastated wildlife. Just last month, WWF released its 2024 Living Planet Report. And the Living Planet Report is an assessment of the health of the planet. So it's really about the state of global biodiversity. And the Living Planet Report shows a 73% decline in the average size of monitored wildlife populations in just 50 years. So 73% decline in 50 years. So that's within my lifetime. It's truly shocking. Additionally, year on year, we're seeing climate records broken. So 2024 is very much on track to be the warmest year on record, beating last year's record heating. And it's putting the health of people and the health of nature at risk. So it's clear that we're facing both a climate crisis, but also a biodiversity crisis or a nature crisis. And until very recently, these two issues were dealt with in complete isolation. So the policy world is now increasingly waking up to the link between the two, and the fact that climate change and the loss of nature are really, they're not separate issues, they're two sides of the same coin, and we can't tackle one without tackling the other. But I guess the good news is that the science is clear. We can still restore nature where it's degraded. We can still limit climate change, but we have to act with urgency, with ambition, with commitment, and also with climate justice. And it's clear that the decisions that are made here in Baku and post-Baku will define our future and the future of our children. So I have a 10-year-old son. He's a very feisty little 10-year-old, and you know, it's really clear to me that he will, you know, that generation will hold us to account. They will not forget. So for WWF, the fight for our world is not just about penguins and pandas and polar bears. It's not an act of charity. It's very much we view it as an act of survival. It's also clear that the polar regions have this absolutely critical role, not only in science, but also in climate action. So that's why we've come to COP29 to launch our new project. And we focus on just one key species in the Southern Ocean, and that's Antarctic krill. Krill is the center of the food web above the ocean floor in the Southern Ocean. It's the reason why humpback whales migrate thousands of miles every summer to spend the summer in the Southern Ocean. And they're also incredibly efficient at drawing down and storing atmospheric carbon. So, it's no reason, it's no surprise that we refer to them as the superheroes of the Southern Ocean. Krill are also targeted by a commercial fishery, which operates predominantly on the Antarctic Peninsula and the Scotia Arc. And the fishery is managed by CAMLR, that's the Commission for the Conservation of Antarctic Marine Living Resources. And it's the CAMLR members who decide on the allowable catch within the fishery and also who decide on the spatial distribution of the fishery. Now, CAMLR, as we heard in an earlier event today, is a consensus-based organisation. So it's currently facing some really big challenges in terms of reaching agreement on a number of issues. That includes marine protected areas. That also includes the spatial distribution of the krill fishery. And those negotiations are influenced strongly by global geopolitics. And as a result of this, progress, as with many multilateral forums, is slow and often hard fought. But what is clear is that when CAMLR is successful, we get conservation wins that are truly of a global significance. Okay, so now I'm going to introduce our first speaker, and I'm very delighted to welcome Dr. Simeon Hill. Simeon is an experienced marine fisheries ecologist at the British Antarctic Survey. Have we lost it? No, still there. Good. Okay. We've lost it on screen here. I think we're still good online though. So Simeon is a very experienced marine and fisheries ecologist at the British Antarctic Survey, and Simeon is also a member of the UK CAMLR delegation. So Simeon, it's clear that the decisions at CAMLR need to be based on the best available scientific evidence. So can you lay out the challenge for us that we're trying to address in our project? Why is it so difficult to estimate krill abundance and distribution?
Thank you, Rod, and good evening to everyone joining us in Baku and to anyone joining us from around the world. So Antarctic krill are the most abundant wild animal species on Earth, and they live in a habitat area of 19 million square kilometers, which is bigger than Russia. And we'll just start by defining terms a bit. So although abundance refers to the number of individuals, it's often easier to think in terms of biomass, which is their total weight, because this is a better measure of how much food is available to predators or how much the fishery is able to catch. Apologies. And we're also talking about distribution, which is how the biomass is spread out over their habitat. It's this wide distribution and its location in the middle of the stormy Southern Ocean that makes it so difficult to monitor krill populations. And we can see this by looking at the areas that have been surveyed using echo sounders on ships. So the red areas on this map show locations of large-scale surveys, and you can see that this only adds up to 20% of the total, or less than 20% of the total krill habitat area. And actually, it's so challenging in terms of logistics and cost to run these surveys that they tend to occur many decades apart, and this is this equates to multiple generations of krill. So it's very difficult to know what happens in the time between these surveys. We can get some information about what happens over shorter timescales because some of the surveys are repeated annually, but these only cover 1% of the krill habitat. These are the survey areas shown in blue. So how do we know the size of the krill habitat if it's not been possible to survey all of it? The main source of information comes from another survey method, which is nets. Scientists have been sampling krill with nets since 1926, and by compiling all of the available information, we can get a good idea of krill distribution in the past. And this is useful for estimating things like the contribution of krill to carbon sequestration. I do apologise, I'm having some difficulty controlling my slides. But a significant problem with this compilation of historic information is that it doesn't tell us anything about krill biomass and distribution tomorrow or in decades to come. The Southern Ocean is changing. It's getting warmer and stormier and losing ice cover, and krill populations are changing too. We can see that there was a big change between the 2000 and 2019 large-scale surveys in the Southwest Atlantic. And over a longer time scale of 90 years, there's been a coherent southward shift in the distribution of krill as the northern edge of their habitat has become less hospitable. As a scientist, I'm interested in, I need to know information about krill distribution so that I can understand krill ecology and their contribution to ecosystem services and how these are likely to change in the future. But distribution and biomass are also critical information for managing the fishery. When the fishery catches krill, it affects the food available to predators like whales and penguins, and it can impact other ecosystem services, including carbon sequestration. There's two main policy tools available to manage these impacts. The first is catch limits. CAMLR has set catch limits for each of the surveyed areas that we looked at earlier. But as we've seen, the distribution can change over time, and so can the biomass in any area. I should say that these catch limits are based on the survey estimates of biomass, so it does affect whether these catch limits are appropriate if the biomass changes. And CAMLR actually aspires to develop what's called feedback management, which is shown on this diagram to the right. And this is effectively to change catch limits regularly in response to changes in biomass. So clearly, information on biomass is critical for setting catch limits. And the second policy tool is spatial protection. You can see that CAMLR has implemented 2 protected areas shown in the light blue on the map, but it aspires to implement a whole network of such protected areas. And these are intended, amongst other things, to protect critical habitats. They could also be used to protect climate refuge areas where species are relatively resilient to the effects of climate change. And again, this requires information on the distribution and how it changes over time. So another demonstration that information on krill distribution is critical for implementing policy. So to summarize, information on distribution and biomass is critical for understanding krill ecology, assessing ecosystem services, managing fisheries, and identifying areas for protection. Traditional ship-based surveys are expensive, both in terms of money and carbon footprint. They're also limited in the information, in the spatial and temporal scales they can provide information for, and they're difficult to maintain. So we need to identify reliable, low-cost and scalable alternatives, and potentially monitoring krill from space can offer just such an alternative. So thank you for your attention, and now back over to Rod.
Thanks. Thank you very much, Simeon. Thank you for that. That was very, very clear indeed. I'm going to move straight into our second speaker. And again, I'm really delighted to welcome an early career scientist this time, so Dr. Kate McGarry. So Kate has very recently completed her PhD in ocean colour remote sensing of zooplankton at the University of Strathclyde in Glasgow in Scotland. So Kate, can we really monitor krill from satellites 100 miles up in the sky? I mean, they're so small. How do we do that? How do we go about doing this?
Thank you so much, Rod. Hello to everyone joining us today. So a little bit of background first. Ocean color remote sensing is the use of ocean color signals to derive information on the types and constituents of things that we find in the water. Different constituents will absorb and scatter light differently, causing variations in the spectral shape and magnitude of reflectance signals received by satellites. From these variable signals, we can actually tell a lot about the biogeochemistry of the water. And in these images, you can see some examples. The top two are two different phytoplankton blooms. On the right, we have this typical green algae bloom with which has the photosynthetic pigment chlorophyll in it, and that is driving the signal there. On the top left, we see a coccolithophore bloom, and coccolithophores are species of phytoplankton that have these calcium carbonate structures called liths. So they're really efficient at scattering light, and they produce these bright white waters. We can also see non-biogenic material as well. We can see things caused by the decomposition of organic matter, which is like this image you can see on the bottom left, or suspended sediments, things coming out from rivers and things like that. I guess the biggest advantage of remote sensing data is that we can look at huge spatiotemporal scales, and that will be great at monitoring long-term change, which is really important when we're facing issues such as climate change. So how do krill fit into this? That's the question of this project. Krill are largely transparent as species, but they do contain carotenoid pigments, and most notably this red pigment called astaxanthin. It's the absorption properties of krill, and most notably the red pigment that we see, that we're interested in characterizing during this project. A few krill on their own wouldn't be able to influence the reflected signals received by a satellite. However, we know that krill form these surface swarms, and these swarms may be strong enough to contribute to ocean color reflectance signals if they are dense enough. These swarms can vary in density and size quite significantly, from these very localized scale swarms that you can see in this image here to aggregations of 100 kilometers, potentially more. These large scale features are the ones that are very hard to sample from ships, and they're our main target for remote sensing. So there has been some previous work done on using ocean color remote sensing for the identification of other zooplankton species. The first was conducted by Bastow et al. 2019, where they discovered these really interesting distinctive red patches from satellite RGB imagery in the Norwegian Sea. At the same time, they saw high concentrations of a zooplankton called Calanus finmarchicus in the water. Calanus, much like Antarctic krill, contain the red pigment astaxanthin. And then building on this study, I, as part of my PhD, developed a color matching technique, which used a combination of in situ measurements of Calanus absorption and radiative transfer modeling to characterize the waters of the Norwegian Sea and produce these order of magnitude estimates of surface Calanus finmarchicus concentrations from space. Since then, we've done some more work to characterize the absorption properties of other species, including some northern krill species. And some of our colleagues over at the British Antarctic Survey did some really interesting preliminary research on looking at the reflectance signals of krill as well. But to further develop all of these techniques that we've developed for other species in other areas, we really need to head down to the Southern Ocean and get our highly accurate absorption measurements of Antarctic krill. So then the hunt for krill is on, and with the aid of Hurtigruten expeditions, we'll be heading down to the Southern Ocean in January to collect krill samples and conduct absorption measurements. wherever we see whales and penguins, we assume krill can't be too far behind. And one way we plan on collecting our samples is by following whale activity in the region. This image here is a really cool image of whales feeding on krill using a bubble net feeding technique, where they trap krill near the surface using these bubble nets to make it easier for them to feed on, which I think is amazing. When we find our samples, we'll collect them using zooplankton nets and then take them straight back to the ship to conduct absorption measurements as quickly as possible. We want our measurements to be really representative of natural populations, so we're only going to pick the best, the healthiest individuals to conduct our measurements. So once we get back to the ship, we'll measure the absorption properties of krill using a point source cavity absorption meter, more affectionately known as a SciCam. I'm not sure how well you can see this video, but this is a video of me placing a smaller species of northern krill into the SciCam. It can be quite a tricky process because you're going one by one. Sometimes they don't want to cooperate, but it's a lot of fun. The SciCam is actually a cavity with highly reflective walls, and that, in conjunction with your isotropic light source, produces a homogeneous light field within the cavity. So if you fill that cavity with anything that absorbs, you can get this very accurate absorption measurement, which is free from scattering error or anything like that. So to obtain our individual krill absorption spectrum, we'll use a method called serial addition, where, as you saw, we add our krill one by one into the SciCam and take an absorption measurement at each step. So this is an example of some data that we did on another zooplankton species. And once we get those absorption measurements, we can look at the relationship between number of individuals and absorption to get our per individual absorption measurement. Once we have that, we can then use our radiative transfer models to model the impact of different concentrations on reflectance signals that would be seen by a satellite. In this example, you can see that increasing the animal concentrations lead to a reduction in reflectance between 400 and 600 nanometers because the organisms are absorbing at these wavelengths. But then how do we relate that to our satellite data? Well, we can compare these modeled reflectance spectra with and without krill absorption to reflectance spectra derived from the satellite and see what combination of constituents or krill are required to get our best spectral match. If krill are required to get optical closure, we can also get an estimate of the concentrations needed to obtain the best result, and then we get our concentrations from space. So just a brief example, here is some preliminary data we collected at the start of this project. In image A, you can see an RGB image of the South Orkney Islands from the 15th of February 2016. What's really interesting about this image is that it overlaps with a British Antarctic Survey-led research cruise. And the extent of that cruise is kind of highlighted by that red dotted box. Throughout that cruise, there was a persistent, fairly large surface swarm of krill that lasted across at least 5 days of the cruise. You can see the cruise extent is actually overlapping with this very interesting large red feature on the image. So when we compared our satellite spectra and our modeled spectra without the inclusion of any krill or astaxanthin component, we get these large differences between what the model is seeing and what the satellite's seeing. As you can see, B. The colors in images B and C represent a measure of difference. So the higher the values, the bigger the differences, right, between the satellite and the model. However, when we compare our satellite data to a model with an astaxanthin component, like we did in C, we could get much better matches between the model and the satellite. In this case, we used our previous Calanus finmarchicus as our proxy for astaxanthin because they both contain similar pigments. And this was a good proof of concept study, so to speak, but we really need our krill absorption properties to actually understand how krill, if it's krill, how they're absorbing and what concentrations we're seeing in the water. When we get those, we can get concentration estimates from images such as these, like this does. So once we have our target Antarctic krill signal, we can then look for it in our data, and we can process time series data of the Southern Ocean across 20 years or more. We have a lot of satellite data to look at, and we can get estimates of surface krill populations on large spatial and temporal scales. And then we can use biomass and habitat suitability models such as these, as well as in situ data, to compare our findings and validate our results. So that's everything from me, just to say thank you very much for listening, and to everyone who's made the initial research possible, including the Norwegian Research Council and NERC. And we're really looking forward to continuing our work in partnership with the British Antarctic Survey and WWF. Thanks very much, and back to Rod.
Thanks. Thank you very much, Kate. That was really mind-blowing science. And if it's successful, this project will provide a new tool for CAMLR to better understand krill ecology and the response of krill to climate change based on large-scale synoptic observations. And the decision makers will benefit from this better data, decision makers at CAMLR, and they'll also benefit from the science-based recommendations on krill distribution and abundance. And in turn, that will help them to manage the rapidly changing Antarctic marine environment, including the conservation of krill and krill predators, including marine spatial protection, and also including the highly precautionary management of the krill fishery in the context of the rapidly warming Southern Ocean. So, it has the potential to be really powerful within CAMLR. So I'm going to open up to questions now. If you would like to ask any questions, either in the audience or online, please do say who you are and your affiliation. But perhaps because I'm at the microphone, I might just ask the first question. So Kate, so clearly we're looking at surface swarms of krill. So what depth can a satellite typically see through the water? And how much of the krill population do you think we can estimate from that satellite data?
Yeah, so the depth the satellite can see to is very dependent on what's in the water around it. So if we've got really turbid waters with a lot of sediment or something like that, or huge chlorophyll bloom, we're not going to see very deep, maybe centimeters, but that's usually the case in very coastal areas. When we're heading out to more open ocean areas, the typical depth range is between 5 and 10 meters. So we're definitely looking at the surface populations. The biggest benefit that we're going to get from satellite data, as we've said, is the spatial and the temporal scale that we can see to, but it also affords the opportunity to have a close look at these surface populations, which can be quite difficult to monitor via nets. Like net sampling usually averages over tens of meters, and things like acoustics, other methods that we can use to look at krill, they also can't resolve that top 10 meters because bubbles cause interference and things like that. So something like using this method to monitor our surface populations in conjunction with all of these net samples, acoustic monitoring, things like that together, we're going to get a really holistic approach to monitoring that will be great when we're looking at how we can move forward with monitoring this species.
Okay, so thanks, Kate. Okay, any questions from the audience? Yes, please.
Can you hear me if I speak out? Shall I take the mic?
Lift up, yeah.
I'll get the microphone a bit closer by. So I'm Jacqueline Stevels from University of Groningen in the Netherlands. I'm a sea ice marine biologist, sea ice biologist, of course, krill is very much dependent on sea ice, and also part of its life stages is associated with sea ice. You can't penetrate a sea ice flow with a satellite. So how do you have any idea what? It's actually maybe a similar question as our chair just asked you. What is the percentage of the population that you will see when knowing that a large part of the krill is underneath the sea ice?
It's an interesting question. I'm not so sure about sea ice in the area that we're looking at so much. But yeah, again, there are going to be limitations to what a satellite can see. Sea ice is one of them. We're definitely not going to be able to penetrate sea ice, or clouds is another one. It can be quite stormy and cloudy in the Southern Ocean, so that's going to be another restriction on satellite data. But again, using satellites to look at sea ice populations under the sea ice is probably not going to be. It's not going to be a valid method. Using another method in conjunction with sea ice, like things like UAVs that go under the sea ice and things like that, would be cool. I think satellite data has its place as part of a holistic monitoring program. I think it's probably using something like net sampling or UAVs or something else for populations under the sea ice are going to be more useful than satellites, I suppose.
Simeon, did you want to add anything to that, or are you happy with that?
Yeah, I think the question was what proportion of the population is under sea ice and would therefore be missed from satellite monitoring. And obviously, this is a difficult question to answer, because we would need some sort of measurement of the proportion of the population that was under sea ice, and it's difficult to sample under sea ice. So this is. It's a reasonable observation. So I can't answer the question about how much of the population is under sea ice. I recently reviewed a paper which had some habitat models, which kind of showed no krill habitat under the major sea ice areas, and I believe that's basically because their source data was from net sampling in areas where it's possible to deploy nets. It doesn't necessarily mean that sea ice is not krill habitat. And I think it is important to recognise that every sampling method we have provides limited information on the krill population. It's either spatially limited, horizontally limited, or depth limited, as Kate has outlined in her answer. So we're not going to end up with a perfect representation of krill abundance across their entire habitat, but actually, as I said in my presentation, the capabilities that may be offered by satellites greatly improve especially the horizontal coverage and the fine temporal coverage compared to traditional ship-based monitoring. So hopefully this will increase our knowledge, but there will still be areas which remain challenging, and maybe other technologies might help us to understand. And I think it's also worth saying that obviously the fishery operates in open water, so the information that is most pertinent to managing the fishery and achieving conservation goals is the areas that are accessible to satellite monitoring. Thank you.
Thanks, Simeon. Did we have any further questions in the pavilion? Yes, please.
I had a couple of questions.
Two questions.
So my name is Ram. I'm with Polar Educators International. I was with Antarctica, New Zealand before COVID, and I did find that different countries focus on their own marine protected areas. So whether it's the Ross Sea MPA with New Zealand or Chile and Argentina. And I find that a lot of the research is fairly isolated to their marine protected areas. So I just wanted to know if your data sets are collaborative and you work with other countries to look at Antarctic krill as a whole? And does it feed into some system, or is it pockets of research? The second thing I wanted to also ask was, as an early career researcher, you would be interacting with other researchers. Are there projects in the same region where collaboration happens? Because I know people who study krill poop. I know people who study Adelaide penguin poop. I know people who study whale poop, all probably in the same region. So if you go out on an expedition, are there people who are focusing on that region, but multiple projects which are maybe interconnected in nature?
Okay, thank you. I'm going to put that to Simeon, if that's okay. Certainly the first question, Simeon, but please do take the second question as well if you feel confident with that one as well.
Yes, thank you. I think you raised some useful issues, and I think one of the important things is that there are constraints on logistics to study the Southern Ocean. Particular countries actually have their own regions of operation, which are sort of defined by a variety of reasons, not least accessibility and the fact that they've established bases in such areas. So there is some sort of geographical separation of research effort, and the real challenge is to bring that together into a more coherent whole. And there are various ongoing large-scale international projects that aim to do that. You asked specifically if there is if the data sets are collaborative. So I'm not sure whether you were talking about the data sets that I referred to. So I could say that the krill-based data set, the compilation of net haul data, is from, I think it was 17 countries have contributed to that. So there is a history of sharing data. Obviously, in order to use satellite data, you need a degree of international cooperation and collaboration, because I think Kate can tell us which countries are responsible for operating the satellites. So this is a small-scale project at the moment, but the whole idea of it is that this is something that will be useful to krill scientists and krill fisheries managers, and the objective of it is to develop the methods and then share those methods widely. And obviously, once we've shared those methods, it will be possible to use them through collaboration, but it will also be possible for other interested parties to use them individually, because what you need is access to satellite images, and then you can go ahead and do your own analysis. I think the final question, if I understood correctly, was, is there an opportunity for collaboration on this specific voyage that Kate is going on to collect krill samples?
Yeah.
So I'll let Kate answer that
Actually,
one.
meet people who are doing similar projects, but with different species.
Okay, yeah. So I think the question, Simeon, was actually this one would be for Kate. So Kate, whether you, on the trip that you're going on, whether there are other scientists doing work on other species such as whales, penguins.
Right. Yeah. I think the particular trip that we're going on, because it's kind of a unique circumstance, heading out with Hurtigruten, we are the only scientists that I'm aware of heading out during the time periods that we're going down on. But I know that there are lots of Hurtigruten expeditions that have other scientists working on other species, whales, a lot of citizen science things going on down there as well, with phytoplankton monitoring and things like that. So I know there are opportunities, potentially not directly on the voyage that we're going on, but with Hurtigruten in general, there are lots of collaborative expeditions going on down there, which are really cool.
Yeah, and certainly on the national operators' vessels, you will have multiple science projects going on at the same time.
I think the RSS Edinburgh is just hitting Antarctica now.
Exactly. Yeah. Alec.
Hi, this is Alec Loon from Scientific American magazine. So I think you guys referred to and I've seen something about this, how krill are moving southward in response to climate change. Is there also evidence mounting that they're moving in response to fishing pressure, or that they may be declining in response to either climate change or fishing pressure in population numbers? And secondly, Rod mentioned that often these decisions about krill management, fisheries management, have a geopolitical aspect to them. A cynical person might ask, what's the use of studying krill distribution if countries are just going to ignore that data and make decisions based on their geopolitical interests? Thank you.
Simeon, if you can host that question, please.
Thank you. Thank you very much for those questions. Two quite challenging questions there. The fishery remains a low impact fishery. Actually, I'm sure you've seen the statistic quoted that catch limits are lower than 1% of the estimated biomass in the area that the fishery operates. So there's been no detectable impact of fishing on krill populations. I should caveat that by saying that, of course, if you take part of a population away, then you reduce the population. But that effect will be very difficult to detect when you have a population that varies massively over time and spatially as well, like krill does, and you're only taking a small amount of it. But I mean, I think a really important thing, although there is no evidence of direct impacts on the krill population in the past, or no detectable impact on krill populations in the past, that as catches grow, or as even if catches remain static, but the krill population in a particular area changes as a result of other pressures like climate change, then those impacts could become more significant. And that is ultimately the goal of fisheries management, is to manage those impacts and make sure that the fishery isn't taking more than the ecosystem can sustain. And that is one of the reasons why we need this information. But that brings me sort of nicely to your second question, which is why bother if all decisions are as a result of geopolitics rather than science. And I think that there is a framework. The CAMWA framework does require scientific evidence. It is supposed to be based on science, and I think it is appropriate that scientists continue to develop that science. It's not appropriate to kind of be defeatist about it and say, oh well, nobody's going to listen to us. There is a demand from the international community, as stated in the CAMWA Convention, that scientific evidence should be available in order to achieve these objectives. I agree that conditions are very challenging at the moment, and an optimist would say that current conditions are not necessarily an indicator of long-term conditions, and hopefully those conditions will improve. Some of my time I'm an optimist. I'll leave it at that. Thank you.
Thanks for that response, Simeon. And perhaps you could just expand on it a little bit, just by describing how we're going to go about getting buy-in from the international science community. So perhaps some of the scientific diplomacy.
Yes, thank you, Rod. That is a good point. That ultimately the decision making in CAMWA starts with scientific working groups that sort of evaluate and agree the science or the scientific evidence and develop advice that can be provided to the Commission, which ultimately makes the decisions. And so there is a lot that can be done to build consensus within that early stage of the scientific working groups and ultimately the scientific committee, basically through doing cooperative science. And I think this does connect with the last question we were asked about cooperation. It is really important that we move between these perceived barriers, these perceived regional barriers of scientific operations, and I suppose between beyond political barriers as well, that, you know, science is being developed to achieve a particular political goal. There should be a shared scientific goal within CAMWA, which is to achieve conservation in the Southern Ocean. And certainly it is our intention in developing this project, developing a key information source which will help inform decision making, that we will do it in a way that is collaborative and hopefully will develop a greater degree of buy-in across the international scientific community. Thank you.
Thanks, Amin. Any further questions from the audience or online? Do we have any questions online? No? Great. Okay, well, I'm going to close down now, but I just want to say a huge thank you to our two fantastic speakers. I also want to thank ICCI for hosting us this evening, and maybe just a closing comment. So, of course, the climate crisis is a global challenge, and it's way bigger than any one species or one particular region of the world. But, you know, we do still have this incredible opportunity here in Baku for many of the world leaders to raise ambition, and for the governments and businesses who are here to meet their promises. You know, we won't forget, and certainly future generations will not forget. So thank you all, and good evening.